Deep within your own cells, right now, a silent war is raging. It's a battle between ancient viral invaders and your body's defense systems. A standoff that can last for your entire life. Today on G Fun Facts Online, we're unraveling the secrets of these viral stowaways, the latent viruses that have mastered the art of hiding in plain sight. When you think of a virus, you probably picture a chaotic takeover. It invades a cell, hijacks its machinery, and churns out thousands of copies of itself until the cell bursts, releasing a new viral army. This is the lytic phase, and it's what causes the familiar symptoms of an acute illness like the flu. But some of the world's most successful viruses play a longer, more patient game. This strategy is called latency. Latency is a dormant phase where the virus switches itself off. It integrates its genetic blueprint into our own DNA or exists as a quiet, stable loop of DNA inside our cells. It stops making new viruses, becoming effectively invisible to our immune system. This isn't just sleeping, it's a calculated retreat. This strategy is used by some very familiar foes, the herpes virus family, for instance. This includes herpes simplex virus, which causes cold sores, varicella zoster virus, which gives us chickenpox and later returns as shingles, and Epstein-Barr virus, the cause of mono. These viruses set up lifelong camp in our cells, like neurons or immune cells, and just wait. Then there are the retroviruses, most famously HIV, the human immunodeficiency virus. HIV stitches its genome directly into the DNA of our immune cells, becoming a permanent fixture. The evolutionary advantage here is huge. By going silent, the virus sidesteps the immune system's initial onslaught, ensuring its long-term survival. It can persist for decades, waiting for the right moment to reactivate and spread. This is why herpes infections are recurrent, and why a cure for HIV has been so incredibly difficult to find. So how do they do it? How does a virus decide to go dark? The answer lies in their genetic code, specifically in sequences called silencer elements, which hijack our own cellular machinery to mute their own genes. Viruses are minimalists. They don't carry their own gene silencing equipment. Instead, they've evolved to brilliantly co-opt our own. They turn our body's most sophisticated gene regulation tools against us. The two main tools they hijack are epigenetics and non-coding RNAs. Let's break that down. Imagine your DNA isn't just a loose strand, but a thread tightly wound around protein spools called histones. This DNA and spool package is called chromatin. The way this chromatin is packed determines which genes are on and which are off. Loosely packed chromatin means genes are accessible and active. Tightly packed, condensed chromatin means the genes are locked away, unreadable. Viruses have become masters at telling our cells how to pack the viral DNA. They do this by directing our enzymes to add chemical tags to those histone spools. Think of them as epigenetic handcuffs. One of the most important silence tags is a chemical mark called H3K27 trimethylation. When this tag is placed on the histones wrapping viral DNA, it acts like a signal, calling in a host protein complex called PRC2. PRC2's job is to compact the chromatin, effectively locking the viral genes in a silent state. It's like telling a librarian to take a specific set of books, the ones for making new viruses, and lock them in a vault. At the same time, viruses also manipulate a second layer of control, non-coding RNAs. Not all RNA in our cells is used to build proteins. A huge portion of it, called non-coding RNA, acts as a regulator. Viruses produce their own special non-coding RNAs to enforce their silence. Some are long, called LNCRNAs, which can act as guides. A viral LNCRNA might physically grab onto that PRC2 silencing complex and drag it directly to the viral genes that need to be shut down, reinforcing the lockdown. Others are tiny molecules called microRNAs, These are like precision-guided molecular scissors. Herpes viruses, in particular, produce a whole arsenal of their own microRNAs. Some of these mRNAs patrol the cell and destroy the very first messenger RNAs that would kickstart viral replication, acting as a crucial brake on the system. Others target the host cell's own genes, for example, to prevent the cell from dying, ensuring the virus has a safe and stable home for years. This combination of epigenetic handcuffs and whispering RNAs creates a robust, multi-layered silencing system that is incredibly difficult to break. While the general tools are the same, different viruses have evolved unique and clever strategies. Let's look at a few of these masters of stealth. First, the herpes simplex virus, or HSV, the cause of cold sores. During latency, HSV lives as a circular piece of DNA called an episome inside our neurons. Here, it performs a brilliant trick. The vast majority of its viral genome, containing all the genes needed to make new viruses, is wrapped up tight in that repressive, silenced chromatin. It's completely shut down. However, one single region remains wide open and active. This is the latency-associated transcript locus, or LAT. The LAT region churns out huge amounts of a non-coding RNA that helps to keep the other viral genes silent. It also produces those microRNAs we talked about, which act as a fail-safe, ready to chop up any accidental messages that might trigger reactivation. But how does the virus keep this one active region safe from the silencing that has spread over the rest of its DNA? The answer seems to be chromatin insulators. Think of them as firewalls. The HSV genome has specific docking sites for a host protein called CTCF. When CTCF binds to the DNA on either side of the active LAT region, it creates a physical boundary, a loop in the DNA that walls off the active region and protects it from the surrounding silence. It's an incredibly elegant way to keep one light on to ensure all the others stay off. Now let's turn to HIV. Unlike herpes, HIV is a retrovirus, meaning it physically integrates its DNA into our own chromosomes. This makes the viral DNA, called a provirus, a permanent part of the infected cell's blueprint. Getting rid of it is like trying to edit a single sentence out of every copy of a billion-page book. HIV latency is mostly controlled by silencing its main promoter, a region called the long terminal repeat, or LTR. It uses the same epigenetic tricks, recruiting silencing complexes to the LTR to shut down transcription. The permanent nature of the HIV provirus has inspired a therapeutic strategy called block and lock, which we'll talk more about later. The idea is to reinforce this natural silencing so deeply that the virus can never wake up again. Finally, let's look at a fascinating recent discovery involving another retrovirus, HTLV1, which can cause a type of leukemia. Scientists found something remarkable. A specific DNA sequence located inside one of the viral genes that acts as a dedicated silencer switch. This intragenic silencer works by recruiting a key host protein called RNAs, which is normally involved in blood cell development. By co-opting this essential host factor, HTLV1 hijacks the cell's own regulatory network to switch itself off. Researchers proved how powerful this was. When they mutated the silencer so it couldn't bind RNAs, the virus became more active and was cleared by the immune system more easily. In a stunning experiment, they even took this HTLV1 silencer and inserted it into the HIV genome. The result? The normally aggressive HIV became more latent and replicated less. This discovery revealed a beautifully simple and direct off switch that a virus evolved to control its own fate. So if these viruses are so good at staying silent, why do people get recurrent cold sores? Or why does shingles appear decades after chickenpox? Because latency is reversible. The sleeper agents can be woken up. This reactivation is triggered by specific signals that tell the virus the host environment has changed. Things like cellular stress, physical trauma, exposure to UV light, hormonal changes, and especially a weakened immune system can all flip the switch. These triggers set off signaling cascades inside the cell that do the exact opposite of silencing. They call in enzymes that strip away the repressive epigenetic marks and add go signals instead. The chromatin opens up, the viral genes are transcribed, and the lytic cycle begins again. This deep understanding of the silence/reactivation switch is now the foundation for a new generation of therapies aimed at, for the first time, actually curing these persistent infections. There are two main, opposing strategies being pursued. The first is called shock and kill. This is the aggressive approach, mainly being tested for HIV. The idea is to use drugs called latency-reversing agents to shock the virus out of its dormant state. By forcing the virus to produce proteins, it makes the infected cell visible to the immune system, which can then hopefully move in and kill it. This is always done while a patient is on standard antiretroviral drugs to ensure any new virus produced can't spread. While it's a powerful idea, the kill part has been tricky. Sometimes the shock isn't enough to get the immune system's attention. The opposite strategy is called block and lock. Instead of waking the virus, this approach aims to reinforce the silence. The goal is to lock the viral genome in a state of such deep latency that it can never reactivate, even without other drugs. This would be a, quote, functional cure. The virus is still there, but it's permanently harmless, like a fossil in our DNA. The leading candidate for this approach is a compound that targets HIV's TAT protein, which is essential for active viral replication. By blocking TAT, the drug not only stops replication, but also triggers a cascade of repressive epigenetic changes that lock the viral promoter into a deep, irreversible silence. The science of viral gene silencing has opened a window into a hidden world inside our very cells. We've gone from just knowing that viruses can go dormant to understanding the precise molecular choreography they use, the epigenetic locks, the non-coding RNA whispers, and the intricate dance with our own proteins. From the clever firewalls of herpes virus to the dedicated off switch of HTLV1, we see just how elegantly these pathogens have evolved to survive. This knowledge is transforming medicine. We are no longer limited to just fighting active viruses. We can now design therapies that target the latent reservoir itself. Whether it's the brute force shock and kill or the subtle subversion of block and lock, we are learning to manipulate the very essence of viral silence. The path to curing chronic infections like HIV or herpes is still long and challenging. But for the first time, we have a detailed map of the silent world these viruses inhabit. By learning how they turn down the volume on their own genes, we may finally learn how to hit the mute button for good, ending the lifelong battle for millions. These silent stowaways have been hiding for millennia, but thanks to science, we're finally starting to read their secrets.